The Immune System, Innate and Adaptive Body Defenses, Chapter 21, by Ashley, Kristen, and Emily. The nonspecific or innate body defenses are the first two lines of defense in the immune system. The first line of defense are the surface membrane barriers. These barriers keep microorganisms such as bacteria and viruses out. The skin contains sebum, which is about pH 5, which inhibits the growth of pathogens and encourages the growth of normal flora. The eyes have tears which contain lysozyme, which is an antibacterial. In the nose and respiratory tract, there is hair and mucous membranes that keep out dirt and dust. In the GI tract, the stomach is acidic and kills most bacteria and normal flora crowd pathogens out. In the GU tract, the males have a long acidic urethra and the females have a shorter urethra and a more acidic vagina, and they are more prone to infections. The next line of defense is your second line of defense, or the nonspecific cellular and chemical defenses. These are phagocytes, which do phagocytosis or cell eating. One type of phagocyte is a macrophage, which attacks at site of chronic infection. They are free macrophages which wander throughout the body, and these include dust cells in the lungs, and fixed macrophages, like Kupffer cells in the liver, which have microglial cells in the brain. There are also neutrophils, which do phagocytosis at site of acute infection. Phagocytes engulf the pathogen, which forms a phagosome, which fuses with a lysosome. And if the pathogen is resistant to acid hydrolases of lysosome, there is a respiratory burst, which is adding more chemicals to oxidize and destroy the bacteria. Then comes opsonization. Add protein and carbohydrate handles to outside a bacteria and makes it easier to destroy. Then you have defensins, which are substances produced by neutrophils which poke holes in the bacteria. Then natural killer cells do not phagocytize. They release chemicals called perforins, which perforate the cell. And they do not need helper T cells. Inflammation helps prevent the spread of harmful substances, remove waste, and kicks off the healing process. It is caused by vasodilation, which causes hyperemia, which is more blood to the area, which makes it red and hot. And it is also caused by increased vascular permeability, which causes exudate, which leads to edema, which is swelling, and dilutes harmful chemicals and brings more oxygen and nutrients to the infected area. Inflammation can also elicit positive chemotaxis, which is the process of attracting more white blood cells. The first step of phagocyte mobilization is more macrophages and neutrophils to the area. Macrophages go to chronic infection and neutrophils go to acute infection. Then, injured cells release leukocytosis-inducing factors, which cause positive chemotaxis to injury, which will bring more macrophages and neutrophils to the area. Then marginization or pavementing occurs. And then diapedesis, which is the process white blood cells use to move from blood vessels into the tissues, occurs. After that, it's phagocytosis. Monocytes are in the blood. When they move out, they become macrophages. The complement system consists of about 20 different proteins floating around plasma. One of these proteins are antimicrobial proteins, which are groups of proteins that attack foreign invaders. Actions of the complement system include direct lysis of pathogens, which is poking holes in the microorganisms by itself using membrane attack complex. Opsonization, which is carbohydrate and proteins which get stuck on the outside of the cell, which help phagocytosis. And it also aids in inflammatory response by increasing inflammation and releasing positive chemotactic agents. The three complement pathways are the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway is initiated by the immune system. This starts the complement system and needs an antigen-antibody complex. The alternative pathway can be started by lack of inhibitors on microorganisms' surface. The lectin pathway can be activated by lectins binding to specific sugars on microorganisms' surface, and these lead to the common pathway. The common pathway is started by the complement pathways discussed in the last slide. In this, the membrane attack complex is formed, which causes direct lysis of the pathogen. Interferons are not virus-specific. They are secreted by cells infected by virus. They warn neighboring cells by blocking protein synthesis and degrading viral DNA. This can be used to treat Hep C genital warts in some cancers. Fever. Your body temperature should be around 98.6 degrees Fahrenheit or 37 degrees Celsius. Pyrogens are the chemicals that cause fever. These are released by macrophages and other white blood cells. Fever can help you fight infection and virus.
The specific body defense is the adaptive defense. It is the third line of defense. The three characteristics of the immune system include antigen specificity, which only works against specific antigens, systemic, meaning the adaptive immune system is active through the whole body, and memory, which is generating a stronger response after seeing the foreign invader for a second time. Cellular immunity is involving your lymphocytes, specifically T cells. It works with cell-to-cell contact to attack the bacteria, the viruses inside the cells, and find cancers.
Humoral immunity is fluid immunity that's generated by antibodies. It is good against bacteria and viruses that are outside of the cells. The float-around fluid makes them more accessible, and they form antibodies in the complement system. Complete antigens are antigens that actually work. There are two characteristics: immunogenicity, that's causing the immunity, and reactivity, that is the immune response working. The antigen reacts with the products of the immune system response. Haptens are molecules that are too small to respond by itself, example of poison ivy. Antigenic determinants are molecules that cause the response. Self-antigens are MHC protein holders on the outside of the cell, and they tell the immune system where they belong. Two types of lymphocytes are T cells and B cells. They are all originated in red bone marrow. T cells become immunocompetent in the thymus, and B cells become immunocompetent in the red bone marrow. They interact with antigens to start a function, and have self-tolerance of any binding to self-antigens that are destroyed and deactivated will attack normal body cells. Macrophages are born as monocytes and act as antigen-presenting cells.
Humoral immune response includes primary immune response and secondary immune response. Primary immune response is the first time your body sees a pathogen. This is the antigen challenge, which is the first encounter between a lymphocyte and the antigen that it fits. This starts clonal selection, which is when a specific antigen binds to specific receptors on a B cell. The antigen attached to a B cell gets help from a helper T, starts clonal selection, which makes memory cells and plasma cells. Plasma cells are factories that make antibodies. These live for 4 to 5 days. Memory cells don't participate in the first attack, but the second time they see an antigen, they can immediately divide into plasma memory cells and cause a secondary immune response. The secondary immune response is immunological memory. The faster and stronger, cells are producing antibodies within hours instead of days. These antibodies are faster, stronger, and there are much more of them. More of humoral immune response contains active immunity and passive immunity. Naturally acquired active immunity is when you get sick, antibodies are produced by your own body, and then you get better. Artificially acquired active immunity is when you are injected with a vaccine. Types of vaccines include live attenuated, dead, and toxoid. A live attenuated vaccine is when you get a weakened or genetically altered dose of the disease. These give lifelong immunity. Dead vaccines, we use the pathogens, stuff that's maybe too dangerous or too risky to be attenuated. You will need other doses of this. Toxoid vaccines doesn't use any part of the organism. They use the toxins that the organism produces. And passive immunity, your naturally acquired passive immunity, you get antibodies through things such as your mother's breast milk. Artificially acquired passive immunity, you get antibodies through a shot. Antibodies are your immunoglobulins. The structure contains 2 heavy chains and 2 light chains. It's a constant region, and the variable region is the antigen binding site. Your classes of antibodies include IgG, which is your second produced, most common immune response. It's a monomer. The primary antibody produced and secondary in a late primary response. Your next one is IgM. It's the first produced. It's produced by plasma cells, good at coagulation, and it's a pentamer. IgA is a secretory antibody. It's secreted from saliva, sweat, breast milk, mucous membranes. It's a dimer. Your next antibody is IgE. It's the rarest. It causes allergic reactions, activates mast cells and basophils, which secrete histamine, and is also a monomer. Your last class is IgD. It is found on the outside of B cells. It's a monomer. The final part of humoral immune response contains mechanisms of antibody diversity and antibody targets and functions. Your mechanisms of antibody diversity include somatic recombination and hypervariable regions. These are genetically coded, and they are how you come up with different antibodies. Your antibody targets and functions include the complement fixation and activation, neutralization, and cross-linking. Complement fixation and activation are how antibodies use classical pathways to turn on the complement system. Complement binds the constant region of antibodies that's bound to target. The complement fixation into target cell's membrane is triggered, which is your membrane attack complex, which equals the lyse cell. Complement cascade, with its C3 and C5a, increase the immune response. Neutralization covers up virus and toxins to prevent harm and allows macrophages to phagocytize. Antibody bind sites on virus or bacterial exotoxin. It blocks so that they can't bind to body cell receptors, which then antigen and antibody complexes are formed and things are phagocytized. Cross-linking is when an antibody grabs 2 or more antigens at a time. This makes the aggregation of cells, makes it easier to phagocytize.
There are 4 types of organ transplants. Autografts are grafts from the same person, for example, a skin graft from a different part of your body. Isografts are grafts between twins that are genetically identical and they have the same MHC. Allografts are grafts between the same species, but they have a greater chance of rejection. And xenografts are grafts from different species, such as a pig heart valve. When preparing for an allograft, it's important to match the MHC and the blood type, and to prevent rejection, the patient is given immunosuppressive therapy drugs. Homeostatic imbalance of immunity includes 2 immunodeficiencies: severe combined immunodeficiency disease, or SCIDS, meaning the patient doesn't have a sufficient immune system, and acquired immune deficiency syndrome, or AIDS, which is caused by HIV and attacks the T helper cells and also shuts down the immune system. Immediate type hypersensitivities, or type 1 reactions, begin within seconds when antigen binds with IgE. Anaphylaxis binds to basophils and mast cells and dumps histamine. Local anaphylaxis includes hay fever, hives, and asthma. Systemic anaphylaxis includes bee stings. Atrophy is spontaneously becoming allergic to something, for example, spontaneously becoming allergic to peanut butter. Subacute hypersensitivities begin within a few hours. Cytotoxic type 2 are reactions with mismatched blood transfusions. Immune complex type 3 are too many antigen and antibody complexes in the blood, for example, lupus and rheumatoid arthritis. Delayed type hypersensitivities, or type 4, takes 1 to 3 days to show up. It is caused by delayed hypersensitivity T cells. Negative reactions include contact dermatitis, such as poison ivy, and graft rejections. An autoimmune disease is the body's trigger for self-to-self attack, examples including lupus, rheumatoid arthritis, and multiple sclerosis.